Osmosis. When a water molecule or solvent moves from a region of high water potential to the region of lower water potential through a semi-permeable membrane down, the concentration gradient is called osmosis. Please remember these highlighted keywords. When you compare with the definition of diffusion, here semi-permeable membrane is included. Let's discuss the basic idea about solution. When one substance dissolves into another, a solution is formed. What is meant by solute? Solute is the substance that is being dissolved. For example, we can call it as salt. What is meant by solvent? Solvent is the dissolving medium. Example is water. So solution is equal to solute plus solvent. Remember, salt sucks the water. Let's see an example. In this beaker, left side has more amount of solvent than the right side. So what is the definition for osmosis? The water molecule moves from the region of higher water potential to the region of lower water potential through the semi-permeable membrane down the concentration gradient. So what does that mean? So the water molecule moves towards the right hand side. So that you can see that the water has moved towards the right side, so the water is very less in the left hand side. Okay, now we are going to discuss about the types of solution. So there are three types of solution. They are isotonic, hypertonic and hypertonic solution. So in the hypertonic solution, you can see that there is a high amount of solute and low amount of solvent. We can call it as concentrated solution, or we can also call it as a lower water potential. If you call about hypertonic solution, the hypertonic solution contains less solute and high amount of solvent. So we can call this as dilute solution. Even you can also call it as high water potential. If you look at this picture, it will be very clear for you to understand. So you can see that here there is a blue color dot one and there is now orange color dotted one. So here you can see that it remains same. So iso means same strength. So same thing is present. In hypertonic solution, you can see that the concentration of the solute is more than the solvent. Same way, if you look at the hypertonic solution, the solvent is more here and the solute is less. Osmosis in plant tissue. When you keep the plant cell in a hypertonic solution, water from the beaker enters into the cell and becomes turgid. Because high amount of water in the beaker is than the cell. This creates a Targa pressure. What is the Targa pressure? The pressure of the cytoplasm pushing against the cell wall. Okay. This pressure prevents too much of water entering. So importantly, you can see that there is a cell wall which prevents the cell from bursting. Okay. When you keep the plant cell in an isotonic solution, the cells remain same because the cells and the beaker containing solution have equal concentration. When you keep the plant cell in a hypertonic solution, which means concentrated solution, so the concentration solution is in the beaker, so there is less amount of water. So what happens? Water from the plant cell leaves out and turn into flaccid. The cells become flaccid because the cell has more amount of water. Okay, so this is a plasmolysed cell. Here totally you can see that the cell membrane is detached from the cell wall. Osmosis in animal tissue. When you keep the RBC in hypertonic solution, water enters into the cell and becomes turgid. So more Targa pressure, due to that the cell gets bursted and becomes a ghost cell. Animal cells doesn't have cell wall, so that it can't hold the Targa pressure. When you place the animal cell in the hypertonic solution, water leaves the cell and becomes shriveled, or we can also say that as granated. So in the isotonic solution, the red blood cells remain same. Importance of osmosis. Due to osmosis, the cell becomes rigid and firm. If not, it turns into wilting form. Secondly, the osmosis helps to provide support and strength. Finally, cell wall prevents water to enter from bursting.